The INFECT project is an EU-funded multinational project. Since 2013, clinical investigators and basic scientists have joined forces to increase the understanding of a group of diseases that are among the most feared infections encountered in medicine, the so-called necrotizing soft tissue infections, NSTI. The leader and coordinator of the project has been Anna Norbe Teglund from Sweden.
Today, despite our modern medicine, severe soft tissue infections cause substantial tissue damage, might be in need of amputation, and the patient may die. The bacteria that causes these infections are commonly found around us. For example, a common cause is strep A that causes sore throat. These patients are in focus in the EU-funded project INFECT. In this project, 14 partners from different countries and from different universities, hospitals and industry has joined forces to tackle these life-threatening infections.
The ultimate goal of the project is to create and improve diagnostic and therapeutic options for the benefit of patients. To incorporate the patient perspective and to help increase dissemination of knowledge to the society, a patient organization, the Lee Spark Foundation, and its founder, Doreen Cartledge, has an important role in the project.
19 years ago, I lost my son, Stephen Lee Spark, from necrotizing fasciitis. He had a strep milleri infection that led to him being eaten away down to his torso, all in 3 days. Necrotizing fasciitis has to have an early diagnosis. After speaking to many medical professionals that said there was a need for a support group for many families affected by this, I decided to start the charity. I have supported thousands of families. All have concerns of early diagnosis due to not being taught properly, and the need for aftercare. And 19 years later, I'm still supporting families.
Since the beginning, many patients and others have joined the efforts of the foundation. One of them is Wayne Page, who himself was severely affected by the disease.
My necrotizing fasciitis case in 2011 was detected early, and I was fortunate when I was treated very quickly and efficiently. I wasn't offered any aftercare with the psychological effects, and through research we know that 75% of patients suffer from mental health issues after their experiences. Through the Lee Spark Foundation, we want to help people overcome this.
Patients such as Wayne are in focus in the INFECT project. Researchers and healthcare professionals from 14 universities, hospitals or industry came together to jointly tackle these life-threatening infections. The project is conducted in the clinic to understand the disease and to collect patient samples. In the laboratories to understand the mechanisms leading to severe tissue damage and death. The gained knowledge is then used to improve diagnosis and treatment of these patients. In the clinic, the involved clinicians collected a large amount of samples and data, providing new insight into how NSTIs develop, manifest and respond to treatment. Ole Hildegård from Denmark has led the clinical part of the project together with several Scandinavian investigators.
Since 2013, the clinical partners of the INFECT study group have created what is now the world's largest prospective observational cohort study of patients admitted with necrotizing soft tissue infections at 5 referral hospitals in the Scandinavian countries, Denmark, Norway and Sweden. All of them capable of providing a multidisciplinary treatment protocol, among those including adjuvant hyperbaric oxygen therapy. Today we have included 409 patients. And here are some of the highlights and our future directions of work.
We found that necrotizing soft tissue infections located to the extremities was associated with infection caused by group A streptococcus, whereas infections located to the abdomen and anogenital area was associated with polymicrobial infection. The infection often started with severe pain and bruising of the skin, and half of the patients had septic shock, especially the patients with group A streptococcus infection. A total of 13% of the patients were amputated, and after 90 days, 18% of the patients had died. Lastly, we found that elevated blood lactate level was associated with 90-day mortality.
We have found that group A streptococcal infections constitute 31% of our NSTI cases. Despite higher rate of septic shock in group A streptococcal NSTI, survival is better than in polymicrobic infections.
We also studied the consequences of having diabetes type 1 and type 2 and an STI. Diabetes constitutes almost 24% of comorbidity in an STI. And our studies so far show that neither type 1 or type 2 or a high BMI are associated with higher 90-day mortality.
We have also shown that certain cytokines and markers of the lectin complement pathway are markers of and predictors of NSTI or necrotizing soft tissue infection severity, as well as markers of death. So, in summary, our large clinical and human biobank material collected by the clinical partners now provide our laboratory partners within the INFECT consortium with the proper tools for developing future and better prognostic tools, improve clinical guidelines, and to be more precise in providing personalized medicine in the future for those patients suffering from severe necrotizing soft tissue infections.
In the lab, the main focus has been on pathogenesis. Several key achievements have been accomplished, both using animal studies, in-depth studies of infected tissue, artificial tissue models, as well as advanced studies of gene expression in the bacteria and the human host.
In order to study these infections in the lab, we developed mouse models that mimic infection and pathology. Through these studies, we have been able to identify host factors that contribute to damage, as well as bacterial components that mediate this pathogenesis.
As some of these bacteria only cause infections in humans, a tissue model resembling human skin was built in the laboratory. The bacteria shown in green readily infect the tissue model, cause damage and spread into deeper tissues. In infected tissue, the bacteria were found to form so-called biofilm. In the biofilm, the bacteria grow closely together, forming a protected coat. Thereby, the bacteria become more resistant to antibiotics and the host immune defense. This may explain why the bacteria are that difficult to treat.
In the recent years, there's been a tremendous development in new biological technologies, which allow to obtain very detailed insights into bacterial biology and to study them directly at the site of infection. These technologies include the so-called next-generation sequencing technologies, which allow rapid and high-throughput studies.
Using next-generation sequencing and all the culture-independent methods, we were really capable to, for the first time, see which bacteria are really involved in this necrotizing fasciitis. And we could see that there were a lot of genera in there that are really difficult to culture, and this was the reason why you did not see them really before. So finally, there were communities, and what we also could see that the bacteria in these communities, they were collaborating. And this collaborating finally results into something similar or symptoms similar to what professional pathogens are doing. So usually you have, let's say, Streptococcus pyogenes that is really doing the necrotizing fasciitis, but here we see that you have a collaboration between organisms, and it looks that there is a similar results going on. But when we look also in detail, we could see that the mechanisms, the polymicrobial communities and the professional pathogens are using, they were different mechanisms involved, and this different mechanism that they're using also results in different results or a different response of the host, or let's say of the patient.
Using a systems medicine approach, the different results were integrated to create models describing key aspects of the disease.
So in this project, we use systems biology approaches, that is mathematical modeling and bioinformatics, to try to predict the factors that can lead to mortality of a patient, and to try to understand how pathogens affect the host and lead to disease.
To transform diagnostic and therapeutic advances into clinical practice, industrial involvement is essential. One diagnostic company has been part of the INFECT project, Cube Diagnostics.
Based on the input of the clinicians and the findings of the scientists, two diagnostic tests had been developed. One for the early identification of causing microorganisms, and the other one for the monitoring of the patient's immune response.
As shown, INFECT is a multidisciplinary project aiming at moving the important field of severe soft tissue infections forward. Significant scientific results have been produced. These may be important steps that point towards future advances for the benefit of patients.
Through the joint action of all 14 partners, substantial knowledge has been gained in the field of these severe infections. We have understood the clinical parts of these infections. We have understood the underlying mechanism of these infections. This knowledge has then been used to advance the diagnosis and the treatment of these patients. What specifically we have understood is how varying these infections are. They are caused by different bacteria, different toxins. The host can have different susceptibility to infection. All of these aspects we need to take into account in order to be able to offer the best, most efficient treatment. This is a field referred to as individualized treatment, and that is what we want to achieve in these infections, because that can save life. And that is now where our future efforts are aiming to introduce individualized treatment in the field of severe soft tissue infections.